Here we've got a subset of our secretion leaders directing proteins to the periplasmic space. We're looking at 39 different secretion leaders as well as just cytoplasmic expression, in this case just in 2 different host strains. And you can see really quickly that the secretion leaders can have a huge impact not only on the amounts but on the quality of the protein. Different secretion leaders have different levels of leader cleavage. So it's something that we want to make sure that we're paying attention to as we are screening our toolbox. In this case, we move forward this one secretion leader number 7, where we were able to, in our 96-well expression format, get about 2 grams per liter of the protein with full cleavage of the secretion leader. Now, I mentioned one of the things that we're working on is really streamlining our recovery, and the thing that would make that most effective is to get the protein out of the cell without having to lyse it. And so we've been focused a lot on extracellular localization so that we can get high quality target protein. It's really important for us to put that through the periplasm first. Most of the proteins that we're making for our partners are disulfide bonded, and disulfide bonds form in the periplasm. So we want to make sure that we get those into the periplasm. Some of these proteins are slower folding. We want to make sure that it's got time to fold before we then allow it to move out into the extracellular space. Also, moving it out into the extracellular space, we can kind of avoid proteases or other things that get released, reduced clipping or other modifications to our protein of interest. Of course, if it's extracellular, we've got simplified recovery. We can just spin down the cells. We've got lower HCPs, no host cell DNA, lower endotoxin, and that streamlines everything. Another potential feature is real-time expression monitoring. So we can just pull samples, spin out the cells, and evaluate the titer without having to lyse everything. So here we're looking at 4 different proteins of varying sizes, so about 15 kilodaltons to about 60. This is 96-well expression data, where we're looking at 3 different extracellular hosts. So these have different host cell modifications that allow for the protein to move out to the extracellular media versus our just standard P. fluorescens host strain. The cell pellet is just, we're going to spin down the cells, separate the cell-free broth or CFB from our cell pellet. The cell pellets are lysed, and we run out the soluble fraction just to see what's left inside the cells. And then look at the extracellular media, the cell-free broth as well. And you can see for these different test proteins, depending on the protein, different host cell modifications are allowing that protein to get out into the cell-free broth. So we're finding that a number of these modifications we're making are allowing proteins of varying sizes, varying types to get out into the extracellular media. Of course, it's really important that the quality of that protein is good, so we can test activity. We also looked at intact mass for each one of these, so you can see for each one of these test proteins, so 2 different antibody derivatives of different sizes, an enzyme, and modified microbial toxin. Each one has really high quality, so we're not seeing clipped products. We're seeing intact mass as expected, and really nice quality out of this 96-well format.